I'm not going to enter into details of each and every one of these topics. In the workshops this afternoon, you will be able to discuss this in detail. But I would like to emphasize that from our perspective, IMI will address important unmet medical needs. And I would let it here. I hope that as many proposals as possible will be submitted for this very exciting topic. We have IMIF, we have also ITRIX. This addresses translational research, which needs management and sharing of lots of data between clinical and preclinical activities. We all know how much data we have accumulated in the last 5-10 years. Nevertheless, it is a moment to exploit it, and in fact, pulling together data, sharing data to come up with some meaningful interpretation and exploitation of what can come from this particular sharing of lots of data. It is a proprietary solutions in companies. J&J is ready to move their Transmart to the public domain. We do welcome this development. And from your perspective, we believe that it is a major opportunity to capitalize and develop this aspect that is so important. The long-term expectation is that ITRIX will see the stable, sustainable platform and service to support academic and commercial translational research across Europe. And we would be very happy if you would contribute to it. Next area, delivering and targeting mechanisms for biological macromolecules. And it isn't a surprise to anyone that biological macromolecules have high potential as therapeutics because they are very specific and can address targets which are not druggable, if you wish, with chemical small molecular drugs. And there are such examples there. The field is taking off, but indeed much more needs to be done, in particular to reach intracellular targets or, for instance, to avoid intracellular degradation. So obviously, the aspects that can be addressed under these topics are quite large and various. Moving forward to the chemical stabilization and delivery of macromolecules. If I would be to equate it to something, is collaboration between different disciplines. You cannot do it without really a good collaboration between drug development teams, molecular and cellular biology, studying the cellular uptake mechanisms, protein and nucleic acid chemistry, manufacturing and characterization of biological macromolecules, and nanotechnologies. Anything else is also that you see fit would be also welcomed. So we believe that the impact on the pharmaceutical industry can be huge because it may open new avenues for developing treatments in currently intractable areas. Moving from one aspect, that one of the delivery, to in vitro predictive biopharmaceutical tools for oral drug delivery. As I was mentioning a couple of minutes ago, oral administration of drugs is preferred in most cases. But this is still a challenge for many targets. Obviously, developing a drug compound that lends itself to oral delivery and optimization of oral drug delivery is still a very empirical process. So what do we expect from you? For a solution, for innovative ways of making this very hard to deliver oral molecules. So targeting, which is the other aspect, and testing, which is still done mostly in animal models, it is resource intensive. So another aspect of it would be to deliver also predictive in vitro tools integrated with in silico models, which obviously will move forward all these fields, and they are urgently needed. Again, as I was mentioning for the other topic, I think that this particular area has very high potential, and it can be accomplished only by combining expertise between different pharmaceutical companies and between the pharma and public. We do welcome also our small and medium enterprise research sector who may come with this very innovative solution answering the very different questions that will be posed by this particular topic. Oh, my formulas have disappeared. But moving from the oral delivery to the next area, the sustainable chemistry, we all know that producing drug substances can be difficult, time-consuming, and very expensive. Sometimes sourcing of raw materials also may be an issue. But nature has found solutions, and we have to learn from the nature, to extract what is to be extracted and transform it in the new generation of drugs that can be used in patients. And I'm putting here some examples. The bark from the endangered Pacific yew tree was used for a time as raw material for a Taxol. Of course, that there are not going to be those trees around. So we need your ingenuity to replace, if you want, what has been done by nature and what we have exploited with something that, based on your ingenuity, can be accomplished in a lab. So what are the objectives? We do welcome, again, in this particular topic, the participation of the small and medium enterprises. We do believe that the objectives here, the novel organic and organometallic catalysts, the process intensification, flow chemistry, biocatalysis, synthetic biology, all these issues can be addressed under this particular topic. The next area is that of the human-induced pluripotent stem cells. These human-induced pluripotent stem cells have opened up many new areas of research, including access to improved in vitro system for disease modeling, drug discovery, and safety assessments. So focus on of topic is patient-derived induced pluripotent stem cells to be used for neurodegenerative or neurodysfunctional diseases, diabetes, and safety assessment.